Hello, I'm Jonathan Bond. I'm a haematologist from Dublin. I'm currently based between University College Dublin and the Systems Biology Ireland Institute and Children's Health Ireland at Crumlin. And I'm going to speak today about a talk I gave at the ESH Congress for Diagnosis and Treatment of Acute Leukemias. So in my talk, I talk about immature TALLs and the ETP subgroup. And I also present some new data, which I'd like to speak about today. So it's about interface acute leukemias. So this is work that I did in my previous job in the Hôpital Necker in Paris, in France, and with the supervision of Professor Elizabeth McIntyre. And this is collaboration with Elisa Laurenti from the University of Cambridge. And really, this project came from an idea to try and implement new bioinformatic tools in order to think about these immature leukemias in a different way. So, as we know at the moment, leukemias are classified mainly by phenotypic resemblance to normal precursors. And that works very well for most leukemias, but immature cases, so that's to say that don't necessarily express all the lineage associated markers or that might have some aberrant marker expression, tend to do less well. And really, in practice, it's necessary to kind of decide whether a patient has acute lymphoblastic leukemia or acute myeloid leukemia, because it's necessary to put them into the appropriate treatment category. But that dichotomy is artificial, really, and in normal blood cell development, we don't have that. So we know that there isn't a strict dichotomy between the lymphoid and myeloid pathways of development, and actually that a lot of the precursors are very pluripotent up to a late stage of development. So we wondered, as our knowledge of normal hematopoiesis has evolved over the past couple of decades, this has really been driven by genomic analyses and the use of new bioinformatic techniques to analyze new subpopulations and to analyze pluripotency. So we wondered whether some of these bioinformatic techniques could be used to think about leukemias a bit differently. So we worked on a project for a number of years actually, and tried a variety of different approaches, but what I speak about in my talk is a specific one called ICGS, which is iterative clustering and guide gene selection. So that's a technique that some people may be familiar with for analyzing single cell RNA sequencing data. So it's a type of clustering that relies on selection of a small number of what's called guide genes that are kind of a minimal set to define coherent gene expression patterns. And essentially, the clustering involves serial iteration and refinement of these guide genes so that you can then cluster based on a minimal number of these key genes. So we use this to analyze a cohort of acute leukemias from Necker. So this was 124 cases, and it was deliberately skewed to have a lot of immature cases, both TALL and AML. And we used this ICGS tool to analyze our cohort. And what we found is that in contrast to traditional methods, instead of having kind of neat boxes of clusters, what we saw was that the ICGS could reveal a spectrum of differentiation. So that's to say we saw a spectrum of cases that were very clearly T lymphoid on one side and clearly myeloid on the other side. And then in between, you had this gradual progression. And these clusters in the middle, which were kind of intermediate clusters, which we ended up calling interface clusters and interface acute leukemias, so IALs for short. And we went on to look in a lot of detail about what these clusters were like, so notably by gene set enrichment analysis. So by GACA, we found that these interface acute leukemias transcriptionally had a lot of activation of different programs that were coherent with multipotent lymphoid progenitors and also multipotent myeloid progenitors, which wasn't too much of a surprise, but the surprise we found was that in fact, within those clusters, that it was actually the cases originally diagnosed as AML that were driving the lymphoid enrichment. So to put that in another way, that these AML cases were actually more lymphoid, in inverted commas, than were the TALL cases. So this is a surprise for us. And we also saw this when you compared them to, for example, mixed phenotype acute leukemias as well, that the enrichment for these lymphoid gene expression programs came from the AMLs. So we went on then to see whether expression of these type of transcriptional signatures was clinically important. So we looked at 2 independent AML cohorts that were in the literature, and what we did was we tried to calculate the transcriptional resemblance to our interface leukemia. So we calculated what's called an IAL score, which is essentially based on the expression patterns that we found in our cohort. And looking at 2 independent cohorts, we found that cases that expressed this signature, so what we called IAL score high, actually had a very poor prognosis in terms of overall survival and also event-free survival. And we were able to look at this in a bit of extra detail in the alpha cohort. So we collaborated with Sylvie Castaigne and Hervé Dompré, who did a lot of analysis for us on this. And we found that this was a robust predictor of prognosis in this cohort, and also that it was an independent variable in multivariate analysis as well, which is quite interesting. So independent of traditional cytogenetic classifiers, the ELN classifier, and also even the LSC17 score, which is a very powerful predictor of prognosis. So this was a sign that the signatures may be prognostically interesting, or at least biologically interesting. And the other thing that we found is that also in these independent cohorts, when we did gene set enrichment analysis again, that also in the independent cohorts, the cases with these high IAL scores, so maybe interface acute leukemia-like, also had enrichment for B-cell gene expression. So this links in with our cohort and the fact that there's maybe the subset of AMLs that are actually a bit lymphoid, if you like, that maybe do less well on AML type treatment. So this is kind of intriguing, and this B-cell signature, it's a preliminary observation, I'd say, but it kind of raises the notion of whether these cases might even be more appropriately thought of as in the kind of mixed phenotype or even lymphoid bracket. And we were interested to see that so our cases had enrichment for RUNX1 mutations, as did the independent cohorts, and we were interested to see there was a good paper from the Sobajo group a couple of years ago that showed that RUNX1 mutated AMLs were responsive to corticosteroids. So this is kind of let's say preliminary evidence that maybe these type of treatment approaches could be considered at some point for these cases. So these are the main data I presented, and I think I'd also add that this notion that immature AMLs and TALLs are biologically similar is not new. So we know that they share a lot of mutations, for example, so in the likes of DNMT3A, ZH2, etc. So people have argued that these cases should be kind of considered in the same bracket before. And during my presentation, I also point people to a very nice review article by Gutierrez and Kenses in the BJ Heme a couple of years ago that outlines a lot of the evidence for this reasoning. And they actually argued for creating a separate category of so-called AMTL, so acute myeloid T lymphoblastic leukemia, whereby these patients could have their own kind of box for enrollment in specific clinical trials where you might be able to determine the correct treatment or maybe correct targeted approaches that would be specifically benefit these rare cases. So I think we'd be very sympathetic to those arguments.